Hey everybody, welcome to week 2 of your distance learning experience. Hope everybody had a chance to look over the material from last week and kind of understand how chromosomes are passed on to sex cells and what the process of meiosis looks like. So again, if you have any questions about anything, let me know, because we're going to kind of keep building on those concepts as we move forward. So this week we're going to look at Mendelian genetics. So this will be our first intro to genetics and the topics that surround these concepts. We'll go ahead and get started here. Chapter 11 in your textbooks, but obviously we don't have those, so that's why this is on here, and we'll go ahead and get started. So a brief history on genetics. There were two emerging theories that occurred early. In the past, people didn't understand how traits were inherited, but there were many guesses based on things that could be observed. So those two theories came about as blending theory and particulate theory. Blending theory, it was thought that offspring were a straight mix of parents, and they had an equal amount of traits from each parent. And then particulate theory stated that traits are inherited as particles, and that those offspring would receive a piece of those particles from each parent. So when scientists came up with these theories, they obviously had an idea that traits were passed on from parent to offspring, but they didn't really know how. So in comes Gregor Mendel. And Gregor Mendel was an Austrian monk who is known as the father of genetics. He discovered how traits were inherited and how chromosomes actually passed those traits on from parent to offspring. So a couple terms you need to be familiar with. Genetics is the study of heredity, and then heredity is going to be the passing of traits from parents to offspring. Mendel did most of his work on pea plants, and I think we mentioned that last week in the last recording that I had, that pea plants had a lot of different traits, but they were very simple traits, and they were pretty easy for Mendel to track. So he did a study on pea plants. Again, they have many traits, and then just some examples listed here are pea plants can be tall, they can be short, they can be purple or have white flowers, and their seeds can be round or wrinkled. And there's a chart or a diagram here on the right side of this slide that shows kind of what those traits look like. So again, here's some more traits. Some of the ones that we just mentioned with seed shape, right? Round or wrinkled. Color can be yellow or green. Flower color, again, purple or white. Pod shapes can be inflated or constricted. We can have yellow or green pods. Again, we can have tall or short pea plants. And then we can have different positions of flowers on the actual stem itself. So you can see flower position being axil or terminal in these cases. An interesting characteristic of pea plants is that they can be both self-fertilized or cross-fertilized, and self-fertilization happens when pollen from the same flower lands on the stigma of the same flower, and then cross-fertilization is also known as cross-pollination, and this is when pollen lands on the stigma of another flower and will create seeds from this process. Some more terminology that you need to be aware of is that of true breeding plants, where these plants always create offspring that look like themselves. And then hybrids are going to be offspring of the true breeding plants. So you'll probably find out that this lesson and this information is going to be based on a lot of new terminology. So do your best to kind of keep up, and hopefully you're writing this information down so that you can have it and not have to rewatch this slideshow or this video over and over again. So some traits are going to be dominant over others, right? So we can look at tall, crossed with short offspring, and then we'll have all tall offspring. So these are going to be hybrids. So in this case, tall is going to be the dominant trait, so a capital T, and short is going to be the recessive trait, and this will be symbolized by a lowercase t. Now, Mendel discovered that each trait was controlled by two factors, so this kind of goes back to that particle theory, but he went a little bit further and actually knew that genes were located on chromosomes, and there are different forms of genes that are known as alleles. So alleles are going to be, again, different forms of genes. Genes are going to be factors that determine your traits, and we already know that a gene is a segment of DNA, and again, those will show phenotype, right? So genotype is going to be what the gene says, and then phenotype is going to be the physical trait, and then genes are going to be located on those chromosomes that are, again, a specific section of DNA. All right, real quick check here to see what we know so far. Questions 1 through 5. Go ahead and jot out the answers on a separate sheet of paper in the margins of your notes that you're taking right now, and see how you did. A little review here in vocabulary. Genotype, we've mentioned before, genotype being what the genes, letters, and the organism has. So in this case, if we're dealing with height of pea plants, big T is going to be tall, little t is going to be short, and there's 3 different combinations that we can have here. We can have big T, we can have big T, or little t in this case, and we'll talk more about that here in a few minutes. And then that genotype is going to determine phenotype. So this is what the pea plant is going to look like. It'll be either a tall plant or a short plant, depending on the dominant trait. And in this case, tall is dominant, so big T is going to be the dominant trait, and that's going to tell if the plant is going to be tall or short. Anytime a big T is present, the plant will be tall. The only time we're going to have a short plant is if it's going to have 2 little t's, and those will be the recessive traits. So the middle trait, the big T, little t, is known as the heterozygote, and again, we'll talk about that more here in a second. But the big T, little t is heterozygous, and even though the little t is present, the big T will mask the appearance of the recessive allele, therefore the big T is dominant, and that genotype will be tall. So I'll go ahead and go through these check for understanding questions with you. If you want to pause the video and try them yourself and then turn it back on to see if you got it right, we'll go ahead and roll through this. So go ahead and pause now if you want, and then I'll get started. So number 1, the passing of traits from parents to offspring is known as heredity. The father of genetics is Gregor Mendel. Genes are located on chromosomes. Every gene is made of 2 alleles. And then 5 here, the organism's outward appearance, such as wrinkled seeds, are referred to as the phenotype. Number 6, the letters, for example, big R, that represent the traits are referred to as genotype. An organism that has 2 different alleles or letters, such as big R, is heterozygous, right? So they are different letters. We have big R, so 2 different alleles, and that is going to be heterozygous. If they were the same, which actually number 7 is alluding to this, so an organism that has number 8, an organism that has 2 of the same alleles or letters, such as big R, is going to be homozygous. So if they're the same, they're homozygous. If they're different, they're heterozygous. So which of the following sets would represent Mendel's parent generation? We're going to look at big R times little r, and that would give us the 3 combinations, big R, big R, little r, and then little r. And then number 10, when 2 different alleles occur together, such as big R, the one that is expressed is dominant. So hopefully you're familiar with these diagrams, or at least you've seen them before. If you know what it is, you can go ahead and pause the video and maybe write down some information regarding this diagram, but I'm going to go ahead and talk about it. The diagram shown below is called a Punnett square, so Punnett square. I will upload a separate video tutorial that will go through simple Punnett squares, and you guys can check that out. It'll go through some animations to show you how a Punnett square is filled out. It will give us genotype and phenotype of two parents. So if we're crossing big R little r and big R little r, the genotypes in the squares are going to be what we actually end up with, right? So the letter actually represents the genotype, and then the flowers in this case are going to be phenotype. A few more slides left here with terminology. What you need to know with Mendel's principles is dominance versus recessiveness, and we've already mentioned this. Some alleles can cover up or hide the other. The one that is expressed is dominant, so more of a review here. If we're looking at different colors, right, in cherries in this case, it looks like we have two choices. The dominant color is going to be red. The recessive color is yellow. So if the genotype big A, both dominant alleles, we're going to have the red phenotype. The middle, little a, this is homozygous recessive. These are the recessive genotypes, and the phenotype is going to be yellow. And then the third option, big A, little a, where we have different alleles, a capital letter and a lowercase letter. The capital letter will mask the recessive trait. Therefore, the dominant trait will show up and the phenotype will be red. Two more slides here. This one being on the principle of segregation. So when a parent makes sperm or eggs, their genes will separate, and the gametes, being the egg or sperm cells, contain only one pair of chromosomes, and we went through this with the process of meiosis last week. Last slide, principle of independent assortment states that each trait is inherited independent of other traits. So we can see multiple possible combinations here to the right of this slide, and each gamete can inherit any of those combination of genes, and you can kind of see how that works with the diagram on the left side of the slide. This is going to set the stage for multiple trait genes that we're going to get into next week. This week we're going to look at monohybrid crosses, so we're going to be looking at single traits and just do the 4 square Punnett squares. Next week we'll get into dihybrid crosses, where we're going to look at 16 squares in a Punnett. So hopefully we can get that figured out through this online experience. Again, I will upload a tutorial that will help you visualize how to create a Punnett square. You're going to have some assignments that will ask you to make Punnett squares or fill out Punnett squares this week. And again, if at any time you have any questions, please let me know. Have a good week, guys.